Welcome back, everybody. I hope that my last attempt to share some of my knowledge with you was informative and somewhat engaging. Today we're going to take a very quick look at the immune system. And as this cartoon demonstrates, really the immune system is the body's natural defense. Every minute of every day, your body is engaged in a constant battle with thousands of microorganisms. So as I just said, immunology is the study of defensive responses to foreign invaders. And as we're finding out, how our immune system develops is really based on something called the hygiene hypothesis. And research has shown that children that are not allowed to go outside and play in the dirt or live in a hypoallergenic environment develop often very weird allergies because their body at that young age is not exposed to any allergens. So when they do come into contact with them later in life, the immune system overreacts and is allergic to them. So to begin our look, we're going to break the immune system down into two divisions. We're going to look at the innate mechanisms and the adaptive specific mechanisms of the immune system. So let's begin with the innate system. The innate mechanisms develop quickly and deal with invaders using limited sets of weapons. Now, these sets of weapons are very powerful, though, because think of how many millions of bacteria your body comes into contact every day, and you're not always sick, even if it feels like it sometimes. So, these include physical barriers like skin and mucous membranes. Now, some of you probably have heard of Crohn's disease, and what Crohn's disease is inflammation caused by the body due to a lack of mucus in certain parts of your gastrointestinal tract. Basically, the mucus does not separate the normal microflora from the adaptive immune system. And the adaptive immune system is constantly trying to clear that normal flora. And as you know, we need that balance between normal flora and, we need that balance between normal bacteria and our own cells. The second defensive mechanism in the innate immune system are secreted enzymes and antimicrobial factors. Then there's inflammation, fever, and phagocytic cells. The adaptive immune system develops after the innate mechanisms break down, and these are directed very specific responses, and we'll get into those in just a few minutes. So the most important fact about the adaptive immune system is that it has memory, and that memory is important because as our immune system sees more and more antigens, it becomes more specific to those antigens and can clear it more effectively. And this is the theory behind vaccines. So what we do is we take something from a pathogen, inject it into our body, and our immune system recognizes it and makes antibodies against this. And amazingly, the more times something is seen by the immune system, the more specific it becomes, so it's able to clear it much more quickly. Unfortunately, microorganisms have developed ways to get around our immune system, and that's why we get sick. And we need to get influenza vaccines every year because influenza mutates at a very high rate, and our body can't mutate at the same rate, or we would just get out of control cancer. The adaptive immune system depends primarily on two types of cells, B and T lymphocytes. So we'll go and break the adaptive immune system down into even smaller categories based on the B and T cell subsets. This first group that we'll look at is the humoral immune response. The presence of an antigen stimulates B lymphocytes, B cells, to produce antibodies, which are glycoproteins that react specifically with the antigen and target these antigens for removal or destruction. And we'll go over how that happens in just a second. I want you guys to think of B cells as factories that are making the weapons for our body. The next group that we'll look at are T cells. This is the cell-mediated immune response. T cells have a lot of functions. They signal to B cells, and that is kind of complicated, but what I want you guys to think of T cells as, they physically attack infected host cells and kill them. Now these can be cancerous cells, transplant cells from, like, say, heart transplant, liver transplant, kidney transplant. If it's not matched correctly, T cells will attack it and kill it. Or cells that are parasitized by viruses or other microorganisms. So Antibodies and antigens. This is really the immune system broken down to its most basic level. Antibodies are bivalent molecules, meaning that they have two areas where they can react with antigens. They're Y-shaped, and their structure looks like this. So here's the structure of an antibody. Up here at the top are variable regions, and these variable regions are what gives the antibodies the ability to react to almost anything. They can react to metals, they can react to toxins, whole bacteria, whole viruses, whatever. But what's really amazing is that the human genome encodes approximately 21,000 genes, but the variable regions of these antibodies you can get variation in the hundreds of millions of different combos. So really almost anything can be recognized. And it's kind of a complex genetic mechanism that encodes for these variable regions, and you'll learn about that in further detail in immunology. Antibodies can be broken down into two different regions. The first region includes the variable regions up here and is called the fragment antigen binding region and consists of a light chain and a heavy chain held together by a disulfide bond. The next region is the constant region, which consists of two heavy chains held together by disulfide bonds. And this heavy chain constant region can change its class into IgE, IgM, and each of those have different functions. Before moving on to talk about antigens, I just want to say two things about antibodies. The first is that with all these hundreds of millions of different combinations of variable regions, sometimes there's antibodies produced that recognize the body as foreign, and some of these autoimmune diseases include type 1 diabetes and asthma. Now, the second thing I want to say about antibodies is that sometimes purified antibodies are used as medication, and one of the latest treatments for Ebola is purified antibodies directed against the virus. In fact, the first two American patients to get Ebola were treated with this new treatment, and both of them survived. So now we'll move on and talk about antigens. Antigens are what are bound by antibodies. They're multivalent, meaning they have multiple areas where antibodies can bind to them. Now, antibodies bind to antigens, but the specific areas on antigens bound by antibodies are called epitopes. And epitopes generally consist of 5 to 10 amino acids or sugar residues. And so as you can see here, there's multiple epitopes on an antigen. So theoretically, you could have tons of different antibodies binding to different locations on the antigen. So if we remember back to a few slides, this is how adaptive memory becomes more and more specific. Each time our body is exposed to this antigen, it recognizes a new epitope. So eventually, all these epitopes could be bound by different antibodies and cleared very quickly by the body. Serology is the science of detecting antigen and antibody interaction, and this can be very important in many medical diagnoses and is useful in pregnancy tests, rapid strep tests, and the detection of HIV. There are two ways that antibodies and antigens can interact, and one of them is precipitation, and the other is agglutination. Precipitation is where a protein or carbohydrate, something that is soluble in solution, falls out. Agglutination is where a whole cell combines with antibodies in this large complex. This large complex comes about because of the bivalent nature of antibodies and the multivalent nature of antigens. So as you can see here, multiple antibodies combine to multiple antigens, and you get this cross-linked large lattice, and eventually those fall out of solution. Before moving on to blood typing, I wanted to show you guys this game. It's a little blood typing game, and it's very easy and it's very informative, and it will help clear up some of the confusion on blood typing. So getting back to the lecture, we're going to look at agglutination, specifically hemagglutination, which is the basis for blood typing. I hope that some of you are aware of your specific blood type, and at the very least, if you're not aware of your blood type, know that there are different blood types within the population. Now, we can figure out what blood type there are based on antibody-antigen interactions. The type of antigen that you have on your red blood cells is the type of blood you have in your body. So here's a first example. So you mix antibody A and antibody B, and you find hemagglutination with anti-A. This would mean that the person has type A blood because the antibody reacts to antigen A. Now think carefully, what type of antibody would be produced in the body? So our body would produce anti-B if we were type A blood because you don't want self-reactivity with your antibodies and antigen. The next example we have is you mix anti-A and anti-B. You get no agglutination with anti-A, but you get agglutination with anti-B. This means that this person has type B blood and has type B glycoproteins on their surface, type B antigen. Again, what type of antibodies would this person's body produce? They would produce anti-A because they don't want anti-B antibodies reacting with B antigen on their red. In this example, you get a hemagglutination with both anti-A and anti-B. So what that means is that antigen A and antigen B are found on the surface of this red blood cell. So thinking about which antibodies are produced in the body, we would say that there are neither anti-A or anti-B because both would be reactive to the antigens on the blood type. This next example, you get no agglutination with either. So following our logic, there would be no antigens present on the blood cells, and so the body would produce both anti-B and anti-A. This person has no glycoproteins and thus is type O. So here's a table of different donors and recipients. So as we said, if you have type A, that means your body produces antibody B. So that means you can accept blood from A or O. Blood type B produces antibody A, and you can accept type B or O. AB, you have no antibodies produced, so that means you can receive blood from everybody because your body's not going to recognize it as foreign. Now, type O, you produce both antibodies in your blood, and you can only receive type O because your antibodies recognize A and B. So type O is known as the universal donor, and AB is the universal acceptor. Those of you that are aware of the ABO blood typing know that there's also a plus and minus, and that stands for the presence or absence of another antigen, the RHD antigen, on red blood cells. An individual who has a blood type of A that is also RHD positive is said to have A plus blood. RHD negative people must be exposed once to the RHD antigen before their bodies will start making antibodies. So theoretically, if you need a blood transfusion, on your first transfusion you can receive plus or minus blood, but after that you will need specifically the negative blood. This is also important in pregnancies. Rh factor is important to an Rh negative woman who is carrying the child of an Rh positive man. This is not important in the first pregnancy, as we said, because antibodies haven't been produced to it. But on the second child, it's very important because the mother shares immunity with the fetus, and antibodies can cross the placenta and recognize the fetus as foreign if it's Rh positive and can result in early termination of the pregnancy. And this is known as hemolytic disease of the newborn. To prevent this, there's a therapy called RhoGAM, and basically the doctor gives the pregnant mother a ton of antibodies, and this fools her body into not making her own antibodies. The antibodies given in RhoGAM do not react to the Rh positive child. Okay, so now we're going to move on to measuring precipitation. And the way that we're going to do that in today's lab is using octoloni gel diffusion. The antigen that we'll be measuring is bovine serum albumin, and anti-BSA antiserum will be the antibody. The way this test is conducted is you have a gel, and in the center of your gel you place your antibody. And around the antibody you have varying dilutions of antigen. The antigen and the antibody will diffuse through the gel, and at some point they will precipitate out. Using this test, we can determine what point the antigen can be diluted and still give a positive precipitation band. In immunology, this dilution factor is often referred to as the titer, even though it is just a unitless factor. Octoloni gels have been used to study how silicone breast implants interact with a woman's immune system. And I looked into this, and what they've found is that women only produce antibodies against silicone breast implants if the silicone breast implant ruptures and the silicone spreads into the body. Now, the body can still recognize implanted medical devices, and the way that it tries to get rid of these is it directs fibroblasts to make collagen capsules around these implanted devices and kind of seal them off from the body, so hopefully they won't cause any damage. And this in itself can be troublesome for the person with the implanted medical device. Now we're going to move on and look at the different white blood cells that are found in your body. And a mnemonic to remember these is never let monkeys eat bananas. So we're going to start with the N, or neutrophils. And these are the most abundant white blood cells in your body. They are phagocytic, and they are the first to respond to an infection or tissue damage. And I'll show you a video of just how fast they move. It's very impressive. They are considered granulocytes because they're kind of grainy under the microscope, and you'll see this in lab today. And the reason they're granular is because they're full of these vacuoles that are full of these enzymes that break down all these phagocytized cells. Okay, I want you guys to think of neutrophils as the first responders of the immune system, the firefighters and the policemen. They circulate in the blood like all other white blood cells, but they move into the tissue when they receive a signal, and they do this very rapidly, as we'll see in this video. So you can see they're moving around, and then they get the signal to swarm to the site of tissue damage. And this is very quickly. This video was over the course of 49 minutes, so you can see just how fast those cells move. The next set of cells we'll look at are the lymphocytes. And remember that the lymphocytes are the B and T cells. The B cells are the factories for the immune system. They make all the weapons, and the weapons are antibodies. And the T cells are like the Pentagon. They are highly specialized, and they kind of direct the show. The next cells that we'll look at are monocytes. And monocytes are similar to neutrophils in that they circulate in the blood and then move into the tissue, but they do this on their own and don't require signaling from infection or tissue damage. When they move into the tissue, they either differentiate into macrophages or dendritic cells. Macrophages are important because they're kind of like the garbage men of the immune system. They pick up all the cellular debris and bacteria and stuff like that floating around in the tissue, and they help start the adaptive immune response. So the last set of white blood cells that we're going to look at are involved in parasitic infection and allergic reactions, and those are the eosinophils and the basophils. And unless you have a parasitic infection or an allergic reaction, you're not going to find very many of these in your blood. But eosinophils and basophils are classified as granulocytes, just like neutrophils, because they are capable of phagocytizing things found in the blood and using these granules full of destructive enzymes to break down their phagocytized objects. And finally, we'll look at the red blood cells. Red blood cells are not really part of the immune system. Really, they're just kind of the transporters of the body. They carry gas to be exchanged and other nutrients to different areas in the body. So, like last time, if anything interests you in today's lecture, please spend 5, 10, or 15 minutes just doing a Google search and seeing what you can learn. And be prepared to share that with your group members and your TAs and Rachel. And thank you again for taking the time to watch this video.